Hello everyone. Today we will be solving biology paper 4, October-November 2043. Question number 1. Plants are sensitive to light. Figure 1.1 shows a sequence of diagrams for two young plant seedlings. The seedlings were first grown in a pot with light from all directions and then placed into boxes. Seedlings A was placed into a box with light entering from one side only. Seedling B was placed in a box with no light. Okay, so the seedling A was placed in a box, light entering from one side, definitely will bend towards the light. So the seedling will bend towards the light. Seedling B would be just taller than the original. Trying to reach light. Complete figure 1.1 by drawing seedling A and seedling B in the boxes to show the expected direction of their growth after 1 week. We completed it. Plants are also sensitive to gravity. Define the term sensitivity. Well, sensitivity is the ability to detect stimuli in the internal or external environment to make appropriate changes. Explain the role of plant hormones in gravitropism. So guys, gravitropism is also a term that can also be used for geotropism. So explain the role of plant hormones in gravitropism or geotropism. Auxin is a plant hormone made by the root and shoot tip only. Auxin diffuses through the plant and more auxin accumulates in lower parts. Auxin stimulates cell elongation and the shoot is able to grow away from gravity. And the shoot is able to grow away from gravity. Next, gravitropism enables plants to survive in their environment. Explain how gravitropism enables a plant to survive. Roots growing down anchors the plant. Roots growing down are more likely to reach water and minerals such as nitrates. Shoots growing up are more likely to reach light. Guys, moving on. Reflexes in animals are also an example of sensitivity. Describe how reflexes in animals differ from sensitivity in plants. Reflexes responds faster. Reflexes uses electrical impulses. Electrical impulses of nerves. Reflex are short-lived. And reflexes are not growth response. The pupil reflex is an example of a reflex that occurs in the eye. The eye is a sense organ. Define the term sense organ. So we just have to define organ and how they can sense it. So group of receptor cells, that can respond to specific stimuli. Moving on. A person ate a meal containing starch and fat. Figure 2.1 shows some events that occur after ingesting this meal. Okay. Absorption of nutrients in the villi, assimilation of fatty acid in the liver, breakdown of large food particles by the teeth, movement of small food particles through the esophagus, secretion of amylase from the salivary glands. Put the events in figure 2.1 into the correct sequence. One has been done for you. Okay, R. So, breakdown of large food particles by the teeth. Okay. So after we break it down, the food must move through the esophagus. So R is number 1. The food must move through the esophagus. Okay. All right. Before the food moves through the esophagus, definitely there will be secretion of amylase from salivary glands. So that must be 2. So this is 3. All right. Absorption of, okay, absorption of nutrients in villi will be the next. And assimilation of fatty acid in the liver will be after that. After absorption, assimilation is possible. So R, then we have T, then S, and then we have P and Q. The stomach lining contains cells that secrete proteins. State the names of 2 cell structures that are involved in making or secreting proteins. So 2 cell structures that are involved in making or secreting proteins. Okay, first of all, we can talk about nucleus, because nucleus provides the code for the protein. Then we can think of ribosomes. Okay, some added structures will be rough endoplasmic reticulum, vesicles, mitochondria, because it provides energy, and etc. State the name of one component of gastric juice and describe its function. One component of gastric juice. Very good question. So one of the major component of gastric juice will be hydrochloric acid. So hydrochloric acid. And its function will be acid creates optimum pH for stomach enzymes such as pepsin. Acid also kills kills harmful microorganisms, such as bacteria. So, yeah, 3 marks achieved. All right. So, if any of you choose not hydrochloric acid, but instead you choose pepsin, then this will be the following answers. If you choose pepsin, then all right, pepsin breaks. Pepsin breaks protein to amino acid. Pepsin and pepsin also digests bacteria. Okay, let's carry on. So this question must be answered like either or if you choose pepsin. There are goblet cells in many parts of the alimentary canal. State the substance that goblet cells secrete. Okay. So goblet cell secretes mucus. Emulsification of fat occurs in the alimentary canal. Describe the process of emulsification of fat. Process of emulsification of fat. Fat is mixed with bile, which physically which mechanically breaks down fat globules into smaller fat globules. This increases surface area of fat globules. State the name of the organ in the alimentary canal where fats are emulsified. Small intestine. Or we can answer duodenum. State the name of the structure in the villi where digested fats are absorbed. Lacteal. Lacteal absorbs digested fat. Figure 3.1 is a part of a food web in a rainforest. Okay. We can see the producers, coconut tree. We can also see the primary consumers. All right. Secondary consumers. In some cases, tertiary consumer as well. Okay, moving on. Complete figure 3.2 using the information in figure 3.1. The table, the first one has been done for you. The number of different producers. Monkey is a secondary consumer. Iguana is a secondary consumer. A frog is a secondary consumer. All right. Anaconda is a secondary consumer. And jaguar is a secondary consumer when it eats the monkey. Okay. So total 5. The number of different trophic levels in the food web. Different trophic levels. So producer, primary consumer, secondary consumer, and then tertiary consumer. So including the producer, primary consumer, secondary, and tertiary, we have 4 trophic levels. How many different trophic levels monkeys feed at? Okay, monkeys, we can see monkeys feed at direct producers. Monkeys feed with grasshoppers. So we can say 2 trophic levels. It feeds on producers and it feeds on insect that feeds on producers. Okay, sometimes anacondas are able to kill and eat jaguars. Using the information in figure 3.1, state the name of the highest possible trophic level of an anaconda that eats a jaguar. Okay. Highest possible trophic level, anaconda that eats a jaguar. Anaconda that eats a jaguar. So basically, if we had anaconda over here, then it would be a quaternary consumer. So definitely 5th trophic level. Explain why the population of anacondas is always smaller than the population of fruit bats in the food web shown in figure 3.1. Use the concept of energy flow in your answer. Okay. So we know that fruit bats are at a lower trophic level. Than anaconda. Okay. The energy transfer along a food chain is efficient, is inefficient. Energy is lost by respiration, digestion of undigested food, movement, etc. So mainly, since energy is lost by these processes of life, that's why most of the energy cannot end up to a very high tropic level, like in that case of anaconda. A giant rat was discovered in a natural rainforest in Vanuatu Island. Scientists wanted to determine if it was a new species. Explain how scientists can use tissue samples to classify this rat. Tissue samples can be used to extract DNA. DNA sequence then can be determined. And DNA then can be compared with other known species. Giant rats eat coconut. Coconuts are grown as monoculture for human consumption. Suggest how monocultures of crop plants can result in extinction of some animals. Monoculture leads to destruction of some habitats. Monoculture requires large quantities of pesticides and herbicides. Monoculture leads to loss of biodiversity and the plants grown in monoculture are often introduced species. So native animals are unable to get food from these crops. Yeast is an example of a single cell fungus. The population of yeast can be estimated by using a light microscope to view a small sample of yeast cell in a modified microscope slide. There is a square chamber which is specific volume cut into the microscope slide and a grid to make it easier to count the number of cells. Figure 4.1 shows the diagram of modified microscope slide. It also shows part of a light microscope with the modified microscope slide filled to the top with a sample of yeast cell. The length of the sides of each small squares in the grid in the modified microscope slide is 200 micrometer. The depth of the chamber is 100 micrometer, so there is a total of 25 squares in the grid. Each small square in the grid has an average of 52 cells. Calculate the concentration of yeast cells per millimeter cube. So, first of all, we need to do the working. 1 millimeter is equals to 1000 micrometer. So, 200 micrometer is equals to 0.2 millimeter. 100 micrometer will be 0.1 millimeter. To find out the volume of each small square, it's going to be 0.2 into 0.2 into 0.1 or 0.004 millimeter cube. Now there are 52 cells. So 0.004 millimeter cube has 52 cells, yeast cells. So thereby 1 millimeter cube has 52 divided by 0.004, 13,000 cells. So the answer will be 13,000 cells per millimeter cube. Okay, guys, we're done with this. Moving on. A small sample of yeast cells and 19 gram per dm cube of sugar were added to a fermenter. The population size of the live yeast cells and the concentration of the sugar in the fermenter were calculated every day for 10 days. The results are shown in figure 4.2 and figure 4.3. Okay. So we can see as the number of days increases, concentration of sugar from 0 to 6 days decreases gradually. From 6 to the ninth day decreases rapidly. And then from ninth day to 10th day, it stops decreasing in concentration. We have a figure 4.2. The population size of live yeast cells, we can see from 0 to 3rd day. 0 to 1st day, we can see a lag phase. And then from 1 to 4th day, we can see a log phase that increases exponentially. Now let's see the question. Draw an X on figure 4.3 to indicate the position of the lag phase. Okay. So figure 4.3 is this one. The position of lag phase should be here. So we drew the X. All right. Complete the line on figure 4.3 to show the expected change in the population size of live yeast cells from day 3 to day 10. All right. Day 3 to day 10. All right. So usually the population follow a sigmoid curve. So there is a initial lag phase. We usually see a lag phase. And after that, we see an exponential phase, followed by a log phase. Okay, so we have this experiment going on until day 10. We can assume since the from day 6 to day 9, concentration of sugar decreased rapidly. So we can assume from day 6 to day 9, we can draw a graph like this. It gradually decreases and followed by that it holds. Okay. Describe and explain the changes in the concentration of sugar in the fermenter over the 10-day period. Okay, so we must describe and explain the changes in the concentration of sugar in the fermenter over the 10-day period. So first of all, description. We know that sugar concentration decreases faster between day 6 and day 9. So we're going to mention that. Between day 6 and 9, there is no change. There is no change in sugar concentration from day 9. Next. We can clearly understand the yeast population affects the sugar concentration. Yeast use sugar in respiration and release energy. The rate of sugar decreases at decreases. The rate of sugar decrease is slow at first because there are few yeast cells. The rate of sugar decrease is slow at first because there are few yeast cells. But as respiration, as anaerobic respiration builds up ethanol, it kills the yeast. So some sugar is left over. Guys, next question. Explain why it is important that there is a tube in the fermenter that allows gases to escape. We know that carbon dioxide is produced during respiration. So we're going to write carbon dioxide is produced by respiration. Allowing the waste gas to escape prevents buildup of pressure. Moving on, guys. This question looks like from reproduction. Figure 5.1 shows a photomicrograph of a sperm cell reaching an egg cell. Okay. Complete the sentence. A nucleus containing a single set of unpaired chromosome in a sperm is called. A nucleus containing a single set of unpaired chromosome in a sperm is called haploid nucleus. State where in the female reproductive system the event shown in figure 5.1 occurs. Okay, fertilization occurs in oviduct. So our answer will be oviduct. Describe what happens from the event shown in figure 5.1 until an embryo is formed. Okay, so basically, in order for this particular sperm to fuse with the egg cell, the sperm is going to release an enzyme called acrosome. So acrosome, a digestive enzyme, Digestive enzymes. The digestive enzyme is released from sperm. Jelly coat is digested. By the enzyme. The fusion of the nuclei leads to fertilization. To form a diploid zygote. The jelly coat hardens after fertilization. Jelly coat hardens after fertilization, and the zygote moves along the uterus towards the oviduct. Towards the uterus. Okay. Mitotic cell division. Mitotic cell division takes place after formation of zygote. The egg cell uses energy stores the egg cell uses energy stores for mitosis. Guys, moving to the next question. Some people use fertility drugs and in vitro fertilization to have children. Outline the use of fertility drugs and process of IVF. Okay. Fertility drugs. FSH, follicle stimulating hormone, or LH, luteinizing hormone, are used as fertility treatment. FSH, FSH is injected during the early stage of menstrual cycle. FSH stimulates follicles to develop and mature. Fertility drugs stimulate the ovaries to produce estrogen. LH stimulates ovulation. Now, the process of IVF. In the process of IVF or in vitro fertilization, eggs are collected and sperms are collected. From the desired candidate. Eggs and sperm are collected either from donor or from the people undergoing fertility treatment. Fertilization takes place outside the body in a petri dish in a dish, in a petri dish. The embryo develops in the dish. At eight cell stage, cells can be collected for screening. The embryo then can be inserted into uterus of the mother for continuing pregnancy. One, two, three. Researchers investigated four different insecticides to determine how effective they were at removing three different species of insects A, B, C from cropland. They tested different concentrations of each insecticide to find the minimum dosage required to remove 95% of each insect population in 2 hours. Each insecticide had the same cost per gram. The results are shown in table 6.1. Species of insect. A. Insecticide 1, 3. Okay. Insecticide 2, 51. Insecticide 3, 55. Insecticide 4, 58. Species B. Insecticide 1, 31, 27, 2, 75. C, 10, 2, 3, 65. Okay. Use the information in table 6.1. Decide which one of the four different insecticide and which doses would be the best choice for removing insect A, B, C from a field of cropland. Explain your choices. Okay. So when it comes to the insecticide, we can see the dosage. Hmm. All right. We can see that insecticide 3 stands out the most because it requires a very low dose. So insecticide 3 and the dosage is 5 milligram per dm cube. Okay. Explanation. Insecticide 3 has lowest dosage required to remove all insect species. So which is both cost effective and less detrimental for the environment. Discuss factors other than doses that should be considered to prevent insecticide damaging the environment. The toxicity of the insecticide. The toxicity of the insecticide. How quickly the insecticide breaks down in the environment. Method of application. Whether it harms non-target. So the toxicity of the insecticide, how quickly the insecticide breaks down in environment, method of application, and whether it harms non-targeted species should be checked before applying an insecticide. Describe the advantages of to farmers using insecticide. The advantages are fewer insect that damages the crop, improves quality of the crop, prevents spread of diseases carried by insect, farmers also use other chemicals on their croplands. State the names of chemicals other than insecticide that are used by farmers on croplands and explain their benefits. So, farmer use magnesium needed for chlorophyll. Farmer uses nitrates needed for making amino acid. Farmer uses herbicides which reduces competition with weeds. Farmer may also use CO2 enrichment specifically in glass houses, for increased rate of photosynthesis. Guys, let's move on to the next question. Insecticides are often made from chemicals that occur naturally in plants. Tobacco plants are one example of this. Suggest why having a natural insecticide is an adaptive feature of plants. Natural insecticide improves the chance of survival for the plant. Describe why people find it difficult to stop smoking tobacco. Tobacco contains nicotine, which is addictive, which is addictive. Stopping when smoker quits smoking, they start getting withdrawal symptoms. State the names of two diseases that can be caused by smoking tobacco. We can name COPD, lung cancer, also coronary heart disease, gum disease, or bronchitis. State the name of component of tobacco that can damage alveoli. Tar. Tar can damage alveoli. Other answers could be dust particles. Guys, that's the end of this particular question paper. See you in the next question paper solving.